Good morning everybody. Now you see, today's topic is same as we were dealing in the last class rather. See, we are dealing with biotechnology, its application in agriculture. Now see, you must know that we were dealing with BT cotton. Okay, so BT cotton is an example where we were dealing with insecticidal protein. Remember, insecticidal protein means what? We were dealing with bacillus, that is known as Bacillus thuringiensis. Okay, so Bacillus thuringiensis, basically it's a bacillus. The name implies that it's a bacteria, but it has the capacity of producing toxins in the form of crystal, and it is not affected to the bacillus because in that condition it is infected. It is in the inactive condition. But when this bacillus is ingested by any other organism and the pH balance inside the body of the bacillus it changes, so this inactive crystals of toxin becomes active and it is very lethal, that causes cell lysis. So these we had done it in the earlier. So commercially that particular state of it is known as cry. Okay, that means this crystal coding part of the DNA, it is known as AC, cry1AC. Okay, now the name derives from the gene, it is coding, that means the crystal coding portion of the DNA is or RNA is known as cry1AC. Now this particular gene that is coding for this has been made into two. See, one is cry1AC that is used for controlling cotton balls. You know, cotton ball, if it is infested by the bacteria or any other pest, you know, this cotton ball will not give the length of the fiber it produces will be short. So that will have lesser commercial value. So if it is engineered with that, the cotton balls will not be infected by any other pest, because any time if the organism takes a bite, it may be a beetle, it may be a weevil or a stem borer, if it takes a bite into this plant tissue, so they will have this active form of toxin and that would make them die. So that is why it is known as insecticidal. Insecticidal means killing. Insecticidal. Okay. Now, and the second one, you see cry2AB. This is also a same variety used in the bacteria, but it is used to control cotton borers. Okay. So this BT cotton because it is a stain of the cotton plant that has been engineered with Bacillus thuringiensis. Now the next part we come here is the pest resistant plant. Now what is pest resistant plant? Pest resistance means any pest cannot infect the plant, because as we all have studied in the earlier classes that excessive use of pesticide, excessive use of DDT, gammaxine, etc. They have got adverse ecological effects. Okay, that means what? It damages the ecological balance. So many plants and animals, they are affected directly by the use of the pesticide. So what is the other option? You know, the best option of pest resistant plant is they use RNA interference. RNA interference means what? This is a very advanced and sophisticated technique in DNA or genetic engineering. What is that? You know that when RNA enters into a cell, it codes. Okay, it codes for a specific mRNA, and that mRNA is translated for protein. So if this condition or the central dogma or the protein synthesis is active within a cell, the cell can survive. Okay, but here you see this RNA interference is actually a cellular defense. What happens? That whenever this RNA enters into the host cell and when it comes for the translation of mRNA, this mRNA it produced, okay, has got a mimicked code. For example, you call it as dsRNA, that is defensive RNA, and what defensive RNA is actually anticodon of this RNA. So that RNA, if DS RNA it goes there, it actually silences this mRNA. That means this specific RNA, if gets attached to this, cannot code for the specific protein. So if that cannot code for the specific protein, the organism cannot survive in the host body. Okay. Now you see Biotechnology of various species, these are nematodes. Okay, they are root infested nematodes. So if this root infested nematode, they are always there. So a plant, if it is engineered using the agrobacterium vector. Okay, so if agrobacterium vector, for example, this is the agrobacterium vector and this is coded by DSRNA. This is the engineered variety. Now when it will enter the host cell. For example, this DNA, this is a particular strain of mRNA. Okay, mRNA, and this dsRNA will produce a complementary mRNA. Complementary mRNA that could completely block the translation of mRNA. It would completely block the translation of mRNA, so no protein synthesis will be there, and tRNA cannot pick up, can't pick any protein. Okay, so this is basically is known as the silencing of the mRNA. So for that reason, the pathogen, pest or pathogen cannot survive in host body. Okay, host body. So that is why this is a very revolutionary technology using genetic engineering to control pests and insects that infest the plant.